Hello everybody, my name is Matthew Taylor and I'm a second year PhD student, and I'm just going to take the time to talk about the project I do, which is exploring the pathogenic mechanisms in Parkinson's disease, but more specifically the regulation of LRRK2 by VPS35. The work that I do is at the University of Dundee in the MRC Protein Phosphorylation and Ubiquitinylation Unit, in which the director and the supervisor of my project is Professor Dario Alessi. I am undertaking a 4-year iCase studentship in collaboration with GlaxoSmithKline under the industrial supervision of Dr. Alistair Reith. Working with GSK, I am given fantastic support and guidance from my industrial supervisor throughout the course of the project, and this allows me to openly discuss new ideas in different directions to take the work forward and maximise the research that we do. So back to Parkinson's disease. It's the second most common neurodegenerative disorder and affects 10 million people worldwide. The clinical symptoms of the disease are well-known motor deficits such as a resting tremor, bradykinesia and structural rigidity, but there are also other non-motor symptoms associated. The pathological hallmarks of Parkinson's are the loss of dopaminergic neurons in the substantia nigra and the accumulation of protein aggregates in these degenerating neurons, which are known as Lewy bodies. But to understand the causes of Parkinson's disease, we look more specifically into the genetics. In fact, 5 to 10% of all Parkinson's cases have a genetic component in which mutations increase the risk of the disease, and there are currently 18 well-identified genes involved in inherited forms of Parkinson's disease. My research tries to understand the interplay between two of these, LRRK2, which was discovered in 2004, and VPS35, discovered in 2011. For both of these, pathogenic mutations cause autosomal dominantly inherited forms of Parkinson's disease. The current understanding of these two proteins is that they work in a common pathway, which is being interrogated through this PhD. So, determining how the biology of these mutations that are seen in these proteins underpins the pathology of Parkinson's disease is key to providing novel therapeutic targets that can be exploited, and it allows the transition of benchwork science to the real world. This isn't just an approach for Parkinson's research, this is an approach for all research. So, to LRRK2, this leucine-rich repeat protein kinase 2. It's a large multi-domain protein kinase and contains protein-protein interaction domains in white, a central GTPase domain in blue, made up of the ROCK-core domain, and a kinase domain in green. And highlighted in red are key, well-established pathogenic mutations that arise in these domains. These pathogenic mutations have been shown to increase the kinase activity of LRRK2, which we measure by its output phosphorylation. So as I said, LRRK2 is a kinase, which means it phosphorylates substrates, and in this case, one key established substrate being the RAB GTPases. Phosphorylation is the process in which a phosphate group is covalently attached to a serine, threonine, or tyrosine residue on its target protein and is a type of post-translational modification that's key in signalling pathways, turning them on and off. RAB GTPases are found at the membranes of organelles and trafficking vesicles and stay at the membrane by a C-terminal carboxyl group shown by the squiggly line in the figure. RAB GTPases are proteins that are thought to be key orchestrators of vesicle trafficking because different RABs associate with different organelles, giving them different identities inside the cell. Following on, another key orchestrator in vesicle trafficking is the retromer complex, which contains our protein VPS35. So what do we know? Well, the retromer complex is made up of two modules, a cargo-selective complex of VPS35, 29 and 26, with VPS35 binding to transmembrane cargo proteins, and a membrane dimer, which brings the cargo selective complex to the membrane. The retromer is primarily found at the endosome membrane, which can be thought of as a sorting hub organelle inside cells. And as the name retromer suggests, it functions in retrograde transport. This is transport either via a recycling pathway of protein cargo to the plasma membrane, or retrograde pathway back to the trans-Golgi network. And this allows protein cargo to be sorted in cells to the correct destination, making it another key player in vesicle trafficking. But the Parkinson-associated VPS35 D620N mutation increases Rab phosphorylation by LRRK2. So back in 2011, the Zimprich group identified VPS35 as a gene linked to Parkinson's, and more specifically, it was this D to N mutation at position 620 in the protein that was discovered. The mutation has been studied and investigated in many pathways to be pathological, but more interestingly for us, it was shown to have an impact in the well-established LRRK2 kinase pathway, which we can see from the data on the right. So when this mutation is knocked into cell lines, and in this case mouse embryonic fibroblast cell lines, it leads to an elevation in the phosphorylation of Rab10, as seen in the box. This is true for both 1 or 2 copies of the mutant gene. And we know this to be through LRRK2, as when you add an inhibitor, the phosphorylation disappears, the inhibitor here being MLI2. But this begs the question, how does this happen? How does the VPS35 D620N mutation regulate LRRK2 activity? Which is shown in the schematic on the left, with the goal of the project being to uncover that question mark. And here are just some examples of the ways in the lab in which we try and answer this fundamental question. So this could be through the use of CRISPR technologies. So being able to genetically alter cell lines we use to knock out or mutate genes and in turn the proteins of interest and analyse the effects. We can also determine interacting proteins of LRRK2 and VPS35 and try and uncover any that mediate this regulation. So for this we use immunoprecipitation and mass spec designed experiments. We can look at the localization of each protein using immunofluorescent techniques and determine how the localization inside the cell changes with the mutations. And another way is identifying mutations in VPS35 that also have this effect on LRRK2. And we can try and see where these mutations lie within VPS35 structure and if they lie in a common domain or binding site to the D620N mutation. So this leaves me to conclude this presentation and reiterate the main goal of my project, which is to uncover the mechanism of VPS35 regulation of LRRK2. And being able to do so may provide new perspectives on how LRRK2 is regulated. Ultimately, this could lead to new targets that we can exploit therapeutically for those Parkinson's patients that have an elevated LRRK2 kinase pathway. And as I mentioned before, this work would not be without the great support and supervision from my Professor Dario Alessi and Dr. Alistair Reith of GSK. So with that, I would like to thank you for listening and gladly take any questions.